Genes are found on chromosomes at specific locations. The orientation of the genes with relation to one another and the distance between them can be determined. The distance is based on the frequency of crossing over between genes on the same chromosome. The closer the genes are to each other, the fewer the crossover events that will occur between them, and vice versa. 3-point test crosses are one way to determine the order of 3 genes on a chromosome, as well as the distance between them. A hypothetical plant has 3 linked genes, all of which control fruit phenotypes. A recessive allele of the first gene, P, determines purple fruit color, where yellow, P+, is the wild-type color. A recessive allele of the second gene, R, determines round fruit shape, where elongated R+ is the wild-type shape. A recessive allele of the third gene, J, gives a juicy fruit, where dry J+ is wild-type. To determine the order of these 3 genes on this chromosome and the distances between them, a test cross is performed between a triple heterozygote and a triple homozygous recessive. The offspring from this cross are sorted and counted. 2 of the phenotypic classes result from no crossing over between the parental chromosomes. These are parental phenotypes. The other 6 phenotypic classes result from either 1 or 2 crossover events between the genes on the parental chromosomes. These are recombinant phenotypes. First, the order of the 3 genes on the chromosome is established. One parent is homozygous recessive, so that parent can only contribute recessive alleles to the offspring. The other parent is heterozygous, with the 3 genes being coupled. That is, all wild-type alleles are on one chromosome and all recessive alleles are on the other. If no crossing over occurs, 2 types of gametes are produced, one with all wild-type alleles and one with all mutant alleles. These are parental gametes. Fusion of these gametes with the triply recessive gamete from parent 2 gives 2 parental classes of offspring, one triply heterozygous with a wild-type appearance, yellow, elongated, dry, and one triply homozygous recessive with an all-mutant appearance, purple, round, juicy. The order of the genes is determined by the progeny class resulting from a double crossover involving the 3 genes. A double crossover event changes the orientation of the gene in the center of the sequence. Since the frequency of a double crossover event is expected to be lower than the frequency of either single crossover event, the double crossover gametes are the least frequent pair found among the phenotypic classes. For this cross, that pair is P plus R plus J and P R J plus. The genes P, R, and J must be arranged in such a way that the center gene switches to give rise to these 2 phenotypic classes. The only possible order compatible with the data is P, J, R. The heterozygous parent, therefore, is P plus J plus R plus PJR. We rewrite the test cross with the gene order we have determined. We call the region between genes P and J region 1, and the region between genes J and R region 2. We also rewrite the test cross progeny based on the gene order. The pairs of recombinant classes derive either from a single crossover in region 1, a single crossover in region 2, or a double crossover involving a crossover in each region. Map distances can now be calculated for the 3 genes. The frequency of crossing over is computed for 2 genes at a time. For the P to J distance, all the crossovers in region 1 are added. The single crossovers appear as P plus JR and P J plus R plus. These are found in classes 3 and 4 for a total of 52 plus 46 equals 98 recombinants. We must also include the double crossovers, since each double crossover includes a single crossover in region 1. The double crossovers appear as P plus J R plus and P J plus R. These are found in classes 7 and 8 for a total of 4 plus 2 equals 6 recombinants. The sum of the 98 single crossovers and 6 double crossovers, 104, is divided by the total number of progeny, 500, and then multiplied by 100% to give a recombination frequency of 20.8%. The map distance between the P and J genes is 20.8. The same method is used to determine the map distance between genes J and R. For the J to R distance, all the crossovers in region 2 are added. The single crossovers appear as P plus, J plus, R, and P, J, R plus. These are found in classes 5 and 6 for a total of 22 plus 22 equals 44 recombinants. As before, we must also include the double crossovers, since each double crossover includes a single crossover in region 2. The double crossovers appear as P plus, J, R plus, and P, J plus, R. These are classes 7 and 8 for a total of 4 plus 2 equals 6 recombinants. The sum of 44 single crossovers and 6 double crossovers, 50, is divided by the total number of progeny, 500, and then multiplied by 100% to give a recombination frequency of 10.0%. The map distance between the J and R genes is 10.0. In summary, a genetic map of the 3 genes P, J, and R has been generated. Map distance between P and J is 20.8, and map distance between J and R is 10.0. Adding these two together gives a map distance between P and R of 30.8.